cheia. Acho que agora vai.
Eu baixei o som para não dar microfonia.
Acho que agora foi, né? Acho que agora...
Eu baixei as duas já.
Agora acho que foi. Oi?
Não, tem que dar o Ctrl P.
Ah, sim. Alt P, né? Windows P, né?
Windows P.
Não, eu coloquei duplicar aqui.
Aquela que tem dado certo.
Os dois estão desligados? Projetar.
Estava ligado.
Agora. Entender.
Agora sim.
Now it's right. Agora vai, né? Vocês escutam bem? Quem está na videochamada também?
Dá para ouvir bem.
Opa,
Tanto
perfeito.
o som quanto a imagem está muito boa.
Ah, perfeito. Então, vou falar primeiro em português e depois em inglês, tá? Então, em primeiro lugar, eu queria agradecer a presença de todos. Vou levantar aqui um pouquinho. Agradecer a presença de todos que estão presencialmente aqui, principalmente a professora Neda. I speak English. Também agradecer a presença do pessoal que está online, né? Professores, professor Cid, né? Com quem a gente vem trabalhando aí com random laser já há alguns anos também. Professora Luciana, professora Regina, os professores aqui todos também. Então, essa palestra vai ser dada pela professora Neda, que veio gentilmente visitar a gente da Itália, tá? E é um projeto de colaboração que eu tenho com a professora já mais ou menos mais de um ano, já, na verdade, né? José, que é nosso ex-aluno de materiais, ele passou um ano na Europa, lá na Itália, no laboratório, e agora a gente está recebendo eles aqui, né? Então, a gente, com muita alegria, agradece a presença da professora. So, thank you for being here.
I understood, by the way.
You know that you are a pop star, Recife too, so...
Nice to meet you all.
Actually, I met you because of Professor Cid, Professor Luciana too, and I think it's an honor to, you are not in person, but I think it's an honor for us and for our research to receive you here. Thank you very much. Take your time. I think I will stay here. I pass the slides for you, okay?
You will, and
Yes,
the
I have you.
pointer, maybe I can
the
use
pointer,
this.
yes.
Can I use
Yes,
this?
yes.
Okay, I
So
can
that's
say
I can say it,
I can
okay.
manage that, yeah.
Okay, I would like this here, and that's your.
Okay. Aspetta un po' complicato. I will. Good afternoon to everybody. It's a great pleasure for me to stay here, to stay with you. I'm thanks and Joseph for these nice days. It's a long time that we are planning this trip, and finally we're here. And I am very happy to visit your labs, to know the professors here and your job, your work, and also the many students that you have. I am a researcher, a senior researcher at the National Institute of Research in Italy. This is the biggest center of research that we have. It's about 100 institutes all over Italy, and we are in the Institute of Complex Systems. And our lab is a photonics lab, and it's situated inside the University of La Sapienza. So we also interact a lot with the academia in that university, and we have students, PhD students, and so on. Ivanir and Davinson asked me to talk about what's our activity, experimental activity in Roma, and I decided to give an overview, and also for students. So some parts are a bit boring for professors and researchers, but in case I go a bit faster. So let's start. I will talk about ordered and disordered macrolasers. And why macrolasers? I love work with them. And the reason is that macrolasers are the smallest lasers that we can have. Because to have a laser, we have to entrap light somehow. And being the wavelength of light around 1 micron in the visible or IR or UV, the smallest size of laser that we can have is on micron scale. So it's very fancy to work with them. You can put everywhere, you can do integration, and also to have this sort of displays. So just this part is mainly, it's fast, but it's mainly for you. So if we talk about laser, so laser is made of 2 components, that is the gain and the amplificator. So if you think about a very simple scheme of laser is something like that, you have a fluorescent material in between 2 mirrors, then you pump your material and the fluorescence goes, then it's on.
Yes. Sorry. It was me. Where? Well, I didn't do anything. What? Here it's okay. It's only the TV. The problem is in the TV. Será que tá instável?
Maybe I can turn this.
Yes, no, but strange. There, it's not. Será que? Oh, well, it kicks it just a little bit, maybe.
No, it works.
Yes, here it works. It's only there. It's the TV, the problem. Oh, jumpy. Yes, it's changing alone. Oh, okay. I don't know what happened.
Talvez se não mexe com o mouse, talvez ele...
Não, mas eu não tava nem...
Não, é a pessoa que ela mexe para mostrar, entendeu?
Really? Maybe if we go with it is not. It's not stable. Color. Okay.
É que aí você está tentando imprimir deles.
Yes.
Aí tem que ser o Windows P. É o Windows P.
Yes, I know.
É o Ctrl P, você vai para a impressão.
Sim. Eu fui sem querer.
É o Windows P.
Pronto, agora foi.
É o Windows.
Ok. Ok.
So to have a laser, we need a gain medium inside at least 2 mirrors, and we excite the pump this gain medium inside the mirrors. The radiation goes forward and backward in between your cavity, and at some point you have the amplification of light, so the laser. And usually the gain material is this is a very simple scheme that you can have because you need inversion of population. So if you consider your the energetic level of your system, of your molecule, of your fluorescent material, when you pump it, all the electrons from the first level, they jump to the third one, and then they relax very fast to the second. But the second relaxation to the lower level is very low in time. So you have an accumulation of electrons in this intermediate level, and then you have a fast decay to the first round and a release of radiation for conservation of energy. So this is the main mechanism how a laser, a big laser, the lasers that we have in our labs work. So you really need again a fluorescent material and the cavity. So again, if we consider the standard laser, the big ones that we have, a cavity has this geometry. And when you have your radiation that travels inside the cavity, you have the constructive interference of radiation. And so you have a batch of standing waves inside your cavity. Standing waves are these waves, different periodicity, and you have all of them inside one cavity. So at the end of the story, what we have is, if we look at the, for example, emission from a cavity, you have all these modes of these resonances inside the cavity here. And to have, for example, a laser, a standard laser some mechanical process or electromechanical process, select just one of these modes, and you have a single mode laser. So this is really basic for students. So for example, if you look at a red laser, probably you have a crystal that emits in a broad band and orange band, and inside the cavity you select just one mode, and you have one selected mode at the red radiation. So we have to transfer all this knowledge on micron sizes. So during the years, a lot of microcavities have been proposed. And you can have still very, very tiny Fabry-Perot, like something like that, cavity, or you can have whispering gallery cavities. That's the topic of today, part of the topic of today. And also you can have a grating of index of refraction or a crystal, photonic crystal. So all these kinds of materials can be a cavity, an optical cavity, or can be an optical laser if you put a gain material inside them. And we are working now a lot on the whispering gallery cavities, so very small, very micron-sized spherical cavities. And they are called whispering gallery because the name is from Lord Rayleigh, who first studied this phenomenon, that is, it was possible inside the St. Cathedral that one person talks to the wall at one point and another person was able to listen to the whisper, even if it was too far, very far, but it was possible to hear the...
Sorry, because yes,
okay.
yes, I
Sorry,
don't know.
it's not my fault.
I'm going to let this because I will try to. Do you want me to pass? If
No, I can.
Yeah,
I can come to sit.
Just yes, I don't know why this is happening actually. Será? It's not. I don't know why this is.
Sometimes it goes.
Sometimes yes.
And
For you,
so.
this way?
Cosa?
Is it bad for you this way or?
I can see here, yeah.
Okay.
And these whispering galleries was the resonances of the acoustic wave inside the dome. So also in this case, when the sound wave travels around this hollow cavity, you have constructive interference around it, and you have the resonances of this big cavity. So it was possible to have to talk one point and to listen very far of that. We can do the same thing in optics in a very, very smaller size. So if we consider, we all know that when radiation passes through two media with different index of refraction, we have the refraction of the radiation, so the deflection of the ray. And it is possible that at some point that you enter in your interface like this and you have a total internal reflection. So all the radiation is reflected on the border. So if you consider this on a spherical geometry, you have light that goes inside. It goes round around your cavity. It cannot go out, and you have an amplification. So it is really also in this case you have constructive interference and the modes of your system. So this is the basic way that it works. Of course, we can solve this. It is possible. So for spheres, everything is solvable.
I think the problem is to
Is
mix
this?
them. I think the mouse is the problem, maybe.
Also, I don't use it.
Okay, let's see. Let's try because it's
I use my finger.
your finger. Okay. Let's see if it helps. I don't know if this is the... Maybe this. Yes, you see, ó, ele tá. Let's go to more configurations. Only to lembrar a localização. This thing there. This is mostrarem somente dois. Aplicar. Ok. And go. Okay. Oh, sorry.
I can use this.
Yes, this.
Not this. Okay.
Yes.
Okay. So we can solve all this for spheres is easy. So we can start from the Maxwell equations. We put all this together. At the end, we all know this, the Helmholtz equation that explains how a wave travels. And so we know really everything, and we can put all this in a sphere and use the Lorentz-Meyer theory that gives you the solution of the Helmholtz equation. So you see that you have the eigenmodes, the resonances are the eigenmode of your system that are dependent on the angles, as you can see there, and of course the radius of your system. So I don't want to go into details because the presentation is long, but just to give you an idea that how these things work theoretically. So when you consider these cavities, you have, like the big one that we have also in the small ones, we have all the modes of the system. So all these waves that travel inside the sphere, you may have different orders due to the condition of resonance that you can see there. So the resonance condition is dependent on the index of refraction, of course, the radius, and the wavelength. And this is what you can see, even if this is a simulation, by eye. So if you consider your sphere, you can see these shiny spots on your system that are the modes. The modes, I mean, the accumulation of radiation in certain points on the surface of the sphere. And the modes can be on the equator or also on different other slices. And we really can analyze theoretically by using these Lorentz-Meyer equations. And so as experimentalists, of course, we want to have a spectrum mainly. So what you're interested are these emission, for example, emission spectra. And you can see that you can have very, very sharp lines that are the modes of your cavity. So it's really like a big cavity, but this is micro-sized. And they are sharp, sub-nanometer, and you have all the modes of your system. And but let's say one of the problems with this kind of cavity is that this radiation that is accumulated on this sphere is really on the sphere. It decays as fast as you go. So if just you go a bit far from the surface, 1 micron, you lose everything. So if you want to use such energy, such an accumulation of light, you have to go very close to it. So a lot of technologies have been explored within the last 20 years, let's say. You can see there, they use fibers, prisms, and the principle is like that. So you can illuminate your sphere with a laser and look at the transmissions, for instance, and use these modes of the cavity. And this has been used a lot for sensing, a lot. I mean, it's about, I think, more than 20 years that people are working on this. And this is because when, for example, if you have an analyte, a protein, that approaches close to your cavity, being everything very sensible to the change of the index of refraction, you see a shift of your resonance. And this is due to the fact that if you consider, for example, here, this is your sphere, your cavity, you have a few particles, a few proteins outside it, and when the pump laser goes inside, instead of doing this part, it does this other part because the interface with air or water is here. So the pathway of the light is bigger, and you see a shift of the resonance. And this is nice for people working in sensing because you have the protein, you look at the resonance, it shifts, so you can quantify the presence of that. And it has been used, the people use not only spheres, other geometries, as you can see there. And let's say in the very, very last 6 years, maybe 7 years, we have started to put also a dye inside the sphere and to do a microlaser. So this is a bit better than the passive cavity because you can have a big signal and you can look at the emission. This is an example that I always show in all the presentation. It's a Nature Photonics publication, very, very nice, where people put these microlasers inside a cardiac cell, and they follow the heart activity with this. So they look at the emission, and just by quantifying the motion, the shift of the resonance, they will be able to follow the activity of the heart, of the cardiac cell. So you see that it's it's very powerful as a technique. And these are all very recent results are published in these last years. People have started to put inside cells and also in vivo. And so we have also started to look at these things during the last years, and we have started to consider the whispering gallery lasers. We have started first with this kind of sample. So we put just a droplet of a dye liquid inside the PDMS matrix, and then we cover it. And we use it as, in this case, just, I will show you later for what we use it. But we just pump it with a green laser that you can see there. This is the sample, and we look at the emission. And by this objective, we divided the emission. One part goes to the CCD for the observation, and one part goes to the spectrometer. And these are the modes. So you can see this. So you can see really by eye the presence of the laser on micron size. In this work, in this specific work, we were able to change the size of the droplets, because the resonance condition is dependent on the radius, on the index of refraction. So we did an array of droplets with different size and with different compositions. So inside the droplet, we put lactosium, proteins, the silica, and these things, just to have a certain emission band. And you can do a barcode and use it as a label. So we proposed it as a smart label in photonics. And then slowly we moved to biosensing during the years. And we have started to, actually we buy these spheres that are polystyrene with dye inside them. And we just put in water, look at the emission. So the geometry is this, we excite with pulse laser, then we look at the emission. This is the typical image that we have, and this is the emission spectrum. And these are the modes. So we look at them and how they behave. These are the typical geometries that we have. This is the excitation, and you can see the detection part. So we have started with Angela. This slide is a bit ruined to the different power, but it's okay. She was my postdoc a few years ago. We have started with her just to do for biosensing. We considered a sphere, and we put a droplet of protein tau. In that case, we were interested in protein tau that is important for Alzheimer. And then we had a collaboration, and we were really able to follow to see how much protein was on this sphere just by looking at the shift of these lines. And we got a good sensibility on this kind of system. And also you can see, you can just put your protein solution, and by increasing the quantity, so different quantities, you have more shift, as you can see. And here, in this kind of measurements, the limit of detection was 400 picogram. That is okay, let's say. But for a photonic sensor, it's okay, but of course, it's still high. Then we moved with Stefano, who is here with us, to do something better, and so for specific detection. So if you think about a biosensor, for example, if you have your blood and you want to do a test, and you want to know what is the quantity of a certain protein, not all of them, you have to be able to detect just that type of protein, not all. So you need a specific binding. Usually in biochemistry, so in the test, that we do, they use antibody and antigen. So this is a typical scheme of also the COVID test that we all made a few years ago. So they put an antibody here, and when the fluid flows, the antigen, for example, that was part of the coronavirus, is attached on the antibody, then a secondary antibody is put, and then you have a fluorophore that gives you the signal, like in this case. So if you have that antigen, that protein in your blood or in your fluid, you see a positive line. If not, you don't see it. But if you want to do tests in laboratory, people usually use these ELISA tests that are based on enzyme-linked immunosorbent assays. And the scheme is more or less the same. They use an antibody on the cover, on the lower part of these wells. And then when you have a certain antigen that is conjugated, is attached, they use the secondary antibody and then a fluorophore also in this case, and they look at the photospectrometer, the signal. So with this, they can say, okay, we look at, for example, protein X, and we know the quantity of that. We wanted to transport all this knowledge on our micron size photonics sensors.
Okay.
And so we have started with Stefano to do this work. He worked a lot in finding the way to cover the spheres with the antibody. In this case, we work for interferon gamma, that is another protein important for the immunological system. So he was able to cover the spheres with antibody, as you can see here, able to catch only the antigen, the interferon gamma one. And so the scheme of the detection and elimination is always the same. Here we just look at the shifts of the resonances when the protein is attached on your sensor. Can I go on?
Can you?
No.
No? No, yes.
Okay. And so this is all the results. I summarize as fast as I can. And so if you to do something very quantitative on that, so you can see that, for example, if we select one of the resonances and we follow the shift during time. So we put a droplet of protein solution, actually he did, and we follow the shift in time as much proteins are attached on your small sphere. And we did this with different concentrations. So you can see here, more particles, more proteins you have, bigger is this shift. And also we demonstrated the specificity. So we tried the same mechanism. So the spheres with the antibody for interferon gamma, what we put lysozyme inside the solution, that is another protein. And we saw that in this case, we have almost not variations. So this demonstrates that only mainly the interferon gamma attacks on your sphere, but not another protein. I'm sorry for that. And at the end, we also obtained the variation of the signal, so the shift of the resonance at variance with the concentration. That is very important for people doing sensing because you want to know how many proteins you're having. Can I go on? Debbie? Can I go on?
Yes.
And we obtained these results, the change of the.
Sorry for that. I think the problem is with the cable. This is, I think, a bad contact.
But now you're fast.
Yes. I will stay.
And so what we did at the end is to compare this standard curve of the sensor with the ELISA one. So this is the chemical, let's say, immunoassay, and this is the photonic one. And you can see that we obtain very similar results. And we were happy because it means that we can use our tiny spheres, tiny microsensors, instead of these chemical immunosensors that require a lot of material, are very long in time, and that they are not label-free, but this is label-free because the signal comes from your sphere and not from any tagging of the protein that you want to analyze. And in these last months, we are also trying to analyze our data, our spectra with deep learning, machine and deep learning, mainly deep learning. And so what he did is we select the resonances group and we look at the shifts But if we are good in doing something with machine learning, we could analyze them fast. So you can train your system, give your data, and at the end you can have a machine that gives you, okay, you have a certain quantity of protein in your system. We did this autoencoder deep learning networks, and this is the very first result. So it seems that the result that we got from deep learning, that are the orange ones, are similar to what we obtained with this calculation and this fitting of the spectra. And so for the future, we are trying. I mean, actually, we did. So we also coupled to the system the optical tweezer technique. With optical tweezer, this is possible optically to catch the spheres and to move them. I don't explain in deep, but if some of you is interested, Stefano knows everything. And so the idea is to take your spheres optically and move it, for example, inside a cell, and to see the change of the index of refraction. So if you have some anomaly in your cell, you would see it.
Pretend I'm not here.
No, it's okay. Sometimes
okay.
I
Yes. Okay.
And also it is possible to use, we can do that, an SLM to do a multiple trap. So you can trap many particles and you can move them in space. And this is something that we have done in the last months, and we are now going to put our spheres inside the cells for biosensing. And just, I don't know if I can touch this. This is something that he made. So really you can move with micron size precision, optically everything, all optical. And so for the moment we are at this stage, but we will put inside cells next week. And so this is the idea. People have started to do that. And that's why I am looking for small spheres with a fluorescence, because if we can put inside the cell and we can use it as really tiny endoscopes, you can put and look at your system. For the moment, what we have done is just we put these large spheres that we have inside the glioblastoma, that is the cell for a brain tumor. And you see it's too big. That's why I said to you, we need smaller ones. And we wanted to study that. And we made some viability test on the cells. So we know that the presence of the spheres doesn't kill the cell, let's say. So the vitality is good. And so we are working on that. I mean, once we have seen that, we see that we can do this. So we're on this direction. And now so this is the part for biosensing, because they asked me to introduce you in case you want to collaborate somehow on that. And then now we go back to an old story, because I have 15 years of experience in random lasers. And so I will show you some old and new results. So as I told you at the very beginning, a laser is something that needs a gain inside the cavity. That's it. But you can also have, instead of the ordered laser that was, for example, the whispering gallery one, a disordered one. Why disordered? Because in this case, you don't have something that goes in an ordered way. The light that is inside the material, the fluorescent, is scattered randomly. And this multiple scattering, it acts as a cavity. So at some point you have the stimulated emission also in this case. And so and the random lasers have been, let's say, introduced first in 1968 by Neda Ghofraniha. This is a theoretical work. It's very nice. And not random lasers. So because at some point we really see a shine, a lasing process on these disordered materials. And then only later, in 1995, thanks to this very, the first paper on random lasers, and we also thank Anderson Gomez and who is one of the authors. And it is the first demonstration of random lasing. So here people put just titania inside a rhodium dye material. They pump the system. First, they look at if the energy, the pumping energy is very low, you have this broadband that is the fluorescence of the system. And if you pump more and more and more, at some point you have the stimulated emission that is this narrow band. And from that moment, many groups in the world worked on random lasers. You can find thousands of papers in that, any kind of system. And of course, I have to say that Brazil contributed a lot, and the group of Professor Sid and also Anderson Gomez, and here you contributed a lot, and we did some work also as in Italy. And so the main mechanism is that you have your disordered material with dye, with pump, and we can look at the emission. The emission is only directional, so we don't have any spatial coherence. We could have, let's say, a bit of longitudinal coherence, but it doesn't matter that it is not a perfect laser, no? Because we can use it for different other purposes. We use many geometries. These are titania in microchannels. These are fibers, very micron-sized, and you can see these are the random lasers here. And what is nice, I mean, nice and difficult, let's say both the things together, is that you never have one type of spectrum. Your spectra here are always disordered. So while in the whispering gallery you could see your lines with distance defined by also by the mathematics, here you don't have. Each time you have a system, you have a different emission spectrum. So you can consider it sort of fingerprint of your material. And the nice thing is that it is really, really random. So we did a lot of work on this. I just showed you a few of them. For example, this is a ceramic, fibers of ceramic, biomimetic, that we used for study the coherence of random lasers, or lithographic systems, or also here is macro channels of paper. So we made the paper lace. And so all this work, let's say, at some point we arrived to this big result, let's say, in 2015. And why it is so important? Because we have this disordered material, this disordered laser. We have started at some point to think about why we don't use it to demonstrate something of fundamental physics. I work in the Institute of Complexity, so my background in phase transitions and disordered material out of equilibrium systems, and so we have started to look at these materials as glassy systems. So this is just for the students. If you want to look at a disordered material, you can imagine something like that. So a disordered material is a material that is never at equilibrium, at thermodynamic equilibrium. It has many states that can be shown. So you can have different pictures of the same material in different molecular compositions. This means that you can have a free energy landscape. So your system is never at the minimum. It's never at equilibrium. You have many metastable states, and your system travels in between them. So this is different if we consider, for example, an ordered material that is a liquid or a crystal, for example. In that case, if you know the temperature, the volume, and the pressure, you have a free energy, energy that describes your system. And in this case, everything is in equilibrium. But things are different if we look out of the complexity of disordered materials. And the spin glass theory is the theory that is the, let's say, that's the mainstream, it's the main theory describing complexity. Now is used everywhere in any kind of investigation, from neural networks to traffic jam, birds fly, computing, or folding of the proteins. So it's very, very, very widespread in applications. I just want to show you a few slides just to give an idea how the spin glass theory. We can
This
describe
part
spin glass
No,
theory in a simple way.
Coloca o lugar que está aí o lugar isso.
And so we have to do a bit of history in this case. Maybe you may like, I don't know. So the Hamiltonian, so the equation that we can use to describe phase transitions is this Hamiltonian of Ising. It comes from 1925, and actually Ising, who was a physician, he introduced it and for his PhD thesis. And he introduced this Hamiltonian, that is for a system made of many spins, so many objects with direction up and down, just two, on a lattice. And the interaction between the spins is given by this coupling J, that if it's positive, you have aligned spins, and if it's negative, you have disaligned. So you can describe, he described in his thesis, PhD thesis, all the phase transitions in ordered system, for example, crystals or liquids, but there was a problem with disordered materials. So from that moment, many physicists have started to consider this Hamiltonian also for disordered systems. That's why, the reason is why, because if you have this lattice of many spins, and you just get closer then, for example, by lowering the temperature somehow, you can, if they are very close, they interact, and you may have frustration. For example, you can consider 3 of them. These 2 are aligned, so have positive J. These 2 are disaligned, so positive and negative, and these 2 is aligned. So you can see that this third one can be both up and down in the same moment. And of course, this gives frustration, because in the system, you may have both the cases. So you can imagine many spins with this kind of frustration, and the system never goes to equilibrium, because it changes in time continuously. So if we go to look at the free energy, you have this landscape, this complex landscape, and the system can go from one valley to another in time. And so it was difficult to have equations to the mathematics to describe this, because usually, if you want to describe the thermodynamics of the system, you need a partition equation, so one Hamiltonian with one minimum energy of free energy. But if you have a lot of them, you cannot do anything. So at that point, Edwards and Anderson in 1975, it is a paper there, a PRL paper. They have introduced what is called the replica trick. So they said, okay, we cannot manage this system with many minima, but we can look at the minima as replica, as copies of the same object. So we can do averages on that. We cannot do physics for all the system. We can do physics for each of them, for example, pressure, volume, and then we average all of them. And this is a trick. So it was, let's say, a clever point. And they introduce here, I don't know what the EBCD is, but it should be the order parameter. That is the Q parameter. It is a multiplication of the spins. And they were able to show this, or maybe in the next slides you can see better the equation. But they were also only able to demonstrate by these ordered systems. So the mathematics was fine, but it worked only for ordered system, not disordered, because something was wrong in that. So this Anderson and Edwards equation was okay, partially okay, because it was not able to describe gaseous systems. And at that point, Giorgio Parisi, not everything is jumped. So at that point, Giorgio Parisi came out with his theory, and he was able to describe finally disordered systems. And to do this, he said that, okay, we have this parameter that is the overlap. It is not a simple number, 1 or 0. It has to be a variable with many numbers, a matrix, and only in this case, so only if we consider this parameter as a matrix with different numbers, we can describe a glassy system. And this was a sort of revolution for people working in glass transition. So this is a picture that he made. He is called the Parisian SAT. He said that the Q value, this parameter, this overlap, I'm sorry for that, is a complex matrix. And if you are at the liquid system, so in the parametric regime, you have everything that is flat. But as soon as you have a phase transition, so you are in the glassy system, you see the clusters of this Q, of this parameter that is used to describe transitions. And so he introduced the P of Q, that is the distribution of the overlap. And he said, okay, we don't have only 0 and 1. We must have all the values in between to see that we have a glass. And then we have started to use random lasers with all this background and to show something. So this is what we observed. So once this is a random laser, a system that is fluorescent, and we pump it with pulsed laser. Each pulse has a different emission, as you can see. So we have started to think, okay, we can think about a replica. So each shot, each image that you can see, it can be considered a replica. And we can use these, we can apply the theories of Giorgio Parisi replica symmetry breaking on that. So if, for example, we take just 4 images, and these are the spectra, the emission spectrum from each of them, you see that it changes. So each one is a replica. Each time you look at your system, it's a different system because you have many minima in your energy landscape. And so we try to do this. I hope you can see the formula, yes. And so we have started to apply the theory of Giorgio Parisi and to calculate this overlap parameter. That is a number. I like this very much because I think Giorgio Parisi was a genius to introduce it because it's just a simple number that says you if a system is in equilibrium or not, just by looking at this number. And before these experiments, everything was proved by simulations and theory, but never people demonstrated it experimentally. So we consider our emission spectra. This is the first result that we got in 2015. When we are at low energy, so the system is not in the glass regime, you can see all the spectra. These are 100 spectra, are quite similar. But when we increase the energy, so we jump to the glassy state, the random glassy state, you see that the spectra change a lot in a random manner. And so we have started to consider, okay, let's consider each wavelength is a spin. And we treat all this with the spin glass theory. We calculated from that the Q parameter, the Parisi overlap parameter. And we calculated the distribution of the Q. So the distribution is important because, as I said you before, only if you have a spread of this Q in between 0 or 1 or minus 1 and 1, you can see that you have a glass. So at the beginning, when you are at the low energy, everything is around 0. So you are, let's say, in the liquid phase or paramagnetic phase. And as soon as you increase the energy, you have this spread of Q, and you transit to the glassy state. And if you consider the maximum, the highest value of Q, you can see that there is a jump that is also the threshold of the laser. So at the threshold of the random laser, we see this jump in the P of Q, in the Q. And this was a very nice result. When we published it, we didn't know that a few years later, it was cited in the motivations of the Nobel Prize to Giorgio Parisi. And also I have to say thank you to all the Brazilian kids, because when we published that, Professor Sid and all the groups here have started to do a lot of experiments on that. And actually, we proved the robustness of the technique in many different systems. And I think the citation that we all have in these motivations is due to a lot of work that has been done here in Brazil. So at the point that we are now, we can consider random lasers as photonic glasses, and we can really use them as experimental that we have in our hands to do statistical mechanics. And I don't know if I can, this is very important because there was no system that could demonstrate it, because people tried with alloys of magnetic systems, they tried with colloidal system, but there was not a physical observable, and it was not possible to calculate the parameter Q of the overlap. We did thanks to the photonics, so we have to say thanks to the photonics. Okay, now it's hard without the slides. Maybe I can put my computer, I don't know.
Maybe if you want.
It's almost finished.
Yeah. This is the best part. What happened?
Essa parte de cima, a figura de cima.
Yeah, no?
How can I do like this, Davidson?
Yes, I think, I don't know how. There it's okay. I think it's something off because of the pen drive.
So I will try to explain with words. So another part of this description of disordered system that was introduced by Giorgio Parisi was what is called ultrametricity. So as I showed you before, in the ansatz, in the matrix with all the clusters, he said, okay, this Q value has to have this structure of clusters, but the structure, the topology is really ultrametric. So what we mean? So the geometry of all the work that we know usually is called metric, because we have the Cauchy inequality that says that if you have 3 points in space, you can have any kind of triangle, isosceles, equilateral, and scaling. And an ultrametric matrix, an ultrametric space is a bit different because you cannot have scaling triangles, but only equilateral and isosceles. This means that, in simple words, that the system that you have has a hierarchical structure, as you can see here. This is a plot made by Parisi in 18. So he said that if you consider 3 points for replica, you see that there are this tree structure, and if you consider these points at the edges of triangle, they are mostly isosceles. So this is an ultrametric structure. That means that you have a tree in the topology of your system. The ultrametricity is a very old story, and it's nothing.
It's nothing.
Maybe it is.
Yes.
So first, it was new, something that it was known from biology. Actually, also Darwin introduced it to describe the original species. So it was something known, but not in physics. And when the spin glass theory was introduced, also in this case it was a novelty, because he said that this structure that we all know from biology is present also in the glass systems, the same one. And what was very nice, there's a paper by him, by Giorgio Parisi and Fiorel, and he really described, by using mathematics, that a glassy system has the same structure of this that you see here. So really you have a tree geometry, a tree topology like that. So when José Vanier came to Italy last year, and he wanted to work with random lasers, he brought his samples that you make here. Now I see how to make them with these soldier powders. And we thought about to do something new. And we thought to maybe to do this, to show this ultrametricity by using our photonic glasses. That has been never done before. And so he made his measurements. Again, we have here the spectra from the system, the samples. At low energy, everything is paramagnetic. The P of Q is centered around 0. And as soon as you have random lasers, so the glassy state, you have the spread of Q. So this is something that we know here. Okay. The first thing that we made, it was, okay, we have access to the Q, to this magical quantity to describe phase transition. And with Davidson and José Vanier, we calculated the experimental matrix of Parisi. So you can see this was the theoretical one introduced by him in 1980. And we did the experimental counterpart by using random lasers. So when you are at low energy, liquid system, all Q are around 0. As soon as you transit into the glassy system in random lasers, all this Q, all the replica make clusters. So I said before that replica are copies of your system. It is not that you have the copies as you want. These copies are organized and are organized in a certain way that is given by this matrix. So they are clusterized, but not spread in a random way as we thought before. And this is, oh, sorry.
No problem.
And this is important. So this is, okay. And another nice thing that we made, actually Davidson worked a lot on that. And inspired by some theoretical work, we said, okay, let's demonstrate this ultrametricity by considering our replica as different points in a geometry, in a topology. Mm-hmm. And he calculated all the triangles made by replica, as you can see here. And we saw that the number of the isosceles triangles increase when you go to the glassy regime. So these are histograms. I don't want to go into the details, but if you want, I can do. So we just calculated all the distances of this triangle, and we show that in the glassy regime, 2 of them are very similar, and the third is very not similar to the other ones. So really, the number of the isosceles increase, while the other one not that much. And this means that your system, this is a true demonstration that you have ultrametricity. So the replica forms clusters, okay, you have the Parisian sats, but these clusters are a topology of isosceles. Mm-hmm. So they have a tree structure, the structure of, this one? Which one?
Yes, the black one, yes.
And this is, I think it's the last one. José Vanier worked a lot on this. Mm-hmm. He was in my office hours and hours at that time. Mm-hmm. And so from the Q values, we made this first with dendrogram. Dendrogram is another way to show the clusterization of the Q. So you can use the matrix, or also you can use this. And you can see also in this case, you can see the transition from paramagnetic liquid to glass or random laser. You see, you have all these clusters and the hierarchical structure that you can see. You can imagine triangles, isosceles. Mm. And so we thought that why not? Let's try to do the landscape of the energy that has been never done experimentally. And it is not really the energy, so we cannot calculate the energy, but we can have the topology of the landscape. So with a bit of manipulation and a lot of algorithms, we succeeded together to obtain this landscape of energy. So you can see these are the minima of the free energy landscape that I showed you many slides before, that was theoretical. So I think that this collaboration is fantastic. For me, it's fantastic. And I just conclude. I mean, I have to thank all of them, Giovanni Evinson, and please come to Italy, come to Roma. We have a lot of nice things to do and to eat. And please, you can. I'm sorry for the presentation because we have technical problems, but we are here until Friday. So if you have any questions.
Thank you very much, Neda. I could say no. I apologize for the technical problems too. Okay, but I think it was very good, and I yes. Thank you very much. Actually, this collaboration for us is also you pushed us. You told us about geometricity that we didn't knew about, and I think we did a very beautiful work, and I think you have beautiful work. So yes. And I think that it is good also for the students to see what we are able to do, that you may go to Rome. Fala em português também para vocês, né? Então, vocês verem o que nós somos capazes de fazer, né? Então, vocês têm uma formação excelente, né? Eu acho que se vocês conseguirem aproveitar isso, aí ninguém segura vocês, né? Agradecer também os professores. Thank you for the professors. Professor Luciana was my former supervisor, but we work together now. Most of the works Luciana participated too, and Professor Sid that is here too, that taught me about replica symmetry and random lasers. Thank you very much for all of you, because without you, we would not work as we do now.
10 years of work.
I think probably something like that.
And the main result, I mean, let's say the main important result come from Italy, Roma, here in Brazil.
Yes.
It's the different groups.
Yes, yes.
I have to say, I mean, also other parts, but we can say that we two are the best ones.
Yes, I think so. Disordered physics.
I think Professor Sid agrees, but I don't know.
Let me.
Sid wants to say something.
I think Sid is also in the call. I don't know if he. I think there is. What happened? I'm in the. I don't know if there was sound now. Estava sem som. Sim, aqui o. Eu não sei se ele está mais aqui ou se ele já saiu, né? Acho que talvez ele. Mas eu agradeço também todos que estão aí presentes, né? Acho que o professor Cid saiu, mas Cid has gone, I think. But
maybe they couldn't follow because.
Yeah, maybe I think the internet was off for one moment, and I don't know if. Oh, it is back here. It is back here. Yes. Yes. They were following with me. Yes, it was working there. Oh, okay. I don't know if there was some technical there, but yes, maybe we call them later. Yes.
No.
You should do it.
Actually, we made something for students a few days ago, maybe, and to explain this replica symmetry breaking. So we showed the different pizzas, and so each of them is a replica of our system. And then we tried to explain this with pizza.
The idea of replicas. Nice. Do you have any question? Vocês têm alguma pergunta? Pode fazer em português, eu traduzo.
But no, please do it. It's no problem.
Qualquer pergunta.
Any kind.
I have a question.
Okay.
I would like to know if an application of random lasers rather than the normal lasers. Is there any?
So in random lasers now, it's about 25 years that we have random lasers. So this part of Paris stuff is new and is, let's say, but there are a lot of applications there, yes. For example, people use also for sensing, because as I told you, if you have your system, it has an emission spectrum that is typical of that. And if you perturb it with something that you have, and you change the index of refraction, and you have to change on the spectrum. There's a work by a Russian group, sorry, I don't remember the name, and they use also for detection of cancer with years ago, for example. So it has been proposed.
So it's already.
It has been proposed in sensing a bit, and it's not that much successful as what we spring out, but it has been proposed, and also for labeling. So if you want to do something, a label, you can do it with, because each material has its random laser emission. So you can consider, for example, a little thing that has a certain emission, you can use a label. We also propose in our project many years ago, you can put, for example, in a very little corner of a paint, Van Gogh paint,
Yeah.
and just to look at the emission, you can see if that is the original one. So in principle, they are not lasers. I mean, we call them lasers, but actually they are not lasers because they don't have any spatial coherence and they have just a little of longitudinal coherence. But this is not a bad thing because you can use it in a different way because you have this broadband emission and they are so you can have very tiny ones, this micro lines, and you can. So there are applications. People propose a lot in these things. They not. I mean, I see the group, the other group, that is the Professor Six group. They use fiber lasers and random fiber lasers.
Yes.
So also, let's say, pure photonics. Now, people are working on a lot of groups.
Yes.
Still.
Daniel,
It's about the energy landscape. I want to know, it takes too long to make that graph. How long does it take in time?
Only the graph.
Yeah, no, the measurement.
The measurement is not the measurement. So the measurement of the emission spectra is not that much long. You just put your sample, illuminate, that's the spectra. Then you have to handle the spectra, but now they have all the software equipment in this. And he worked a couple of months, I think, to get the landscape. But now we know how to do it. So now it's fast.
Very fast.
Now it's fast, yes. But at the beginning, we didn't know how to use this data.
Yes.
And nobody did it. So we
began from
we had a lot of
the equations.
Yeah, we started from the equations. I read all the papers by Parisi in 20 years, something like that. But at the end, we succeeded last year. So
The good thing is that she had all the equations, we had the data that were measured there, and we had to work a little bit for some months, I think, to try to apply the equations to our data. But I think
But now
demonstrated.
we have a lot of. I think we can still do a lot of things.
Yes.
Which kind of computer programming?
No.
Like this one.
Program.
MATLAB.
MATLAB or Python.
Or both.
Yes, or both. Yes.
But it is not. It doesn't require computing.
No. I need. Yes. No.
Once you can organize the data in the way you want, you can do everything.
Yes.
Because for Parisi, this famous Q, the magic Q value was just a number 0, 1, and something in between 0, 1. But we can calculate it from experiments. So we know that it's 0, 0.1, 0.5, and so on. We can really quantify it experimentally. And so we can describe the system in a very precise way.
Yes. And as she said, I think at least recently, only random lasers were able to, or we could calculate Q values only for random lasers. And it was introduced by Neda's group.
We have started in random lasers, and then there are few works in nonlinear optics too.
Yes.
In propagation of waves, also some of my colleagues did in Roma. So, I mean, if you have a system that it has fluctuations, and if from these fluctuations you can find a sort of spin, you can do on that system also. You can apply this to that system too. The nice thing in photonics is that we measure it, but you can also do it for nonlinearity. You can do that.
That this is quantum computing.
Quantum computing, I am not a fan of quantum computing, but now all this...
Zero and one and in
Yeah,
between.
yes,
Yes.
it is now, a lot of groups are working on this because people are working on the Ising machine and the Boltzmann machine that are all based on these theories. And they are used this so they don't use random laser, just use a spatial light modulator. They modulate the light and then use the output. So it's a bit easier because we have a magnetic material, pamphlet, but you can do also with an SLM, just modulate radiation.
More questions? Mais perguntas? Pode fazer em português também, não sejam tímidos, né?
This is about the whispering.
Whispering gallery.
Could you comment to limitations and the advantage of whispering gallery mode comparing with the traditional resonators, please?
Traditional resonator, you mean the big one?
The traditional, the difference
So
in
the main advantage is that you have something that is on macro scale. So it's completely different. It's the same thing that I said before. You cannot see a random laser or a whispering laser as a laser. You cannot do that laser as something because we buy, they build, they work, whatever. They are okay. But when you have these very, very tiny lasers, so in this case 10 micron or 30 micron, the good thing is that you can put whatever you want. So, for example, if you want to do an investigation in a cell, you can insert inside your cell, remove it in this case, and see what happens. But you can do this with another traditional laser.
Okay.
So, and the signal is very reproducible. So this, the whispering gallery lasers, you really have a nice signal, and you can, it's stable, time, it's reproducible, and so you can use it. And the problems are always the same. You have to be able to do nice samples, to put nice gaining materials, to avoid bleaching, and so careful with the handling of the samples. And also in this case, data analysis is important because Stefano made a great job last year because there's a shift that I showed you and I went a bit fast on that, but are very, very tiny. So to have a nice result and to see, okay, we have the presence of the protein, he analyzed a lot of data and did a lot of fitting procedures, and at the end we succeeded into this. That's why now we are trying to do deep learning of that. So to use algorithms or neural networks to analyze the data to be faster and more robust. So these are the limitations. So you have to be careful with the samples and also with the data that you have.
Questions? I had only. Okay. Please.
You show in one part and the theory of Darwin. What is this relationship with part of this work?
Yes. So when I talk about ultrametricity, so ultrametrics system topology is the one that has a structure of a tree. So a hierarchical structure that it was known in paleology, in cryptology, before physics. And when physicians started to work on that, they find the same thing in spin glasses, the same structure, and we have the mathematical description of that. So the relation is that in 1 century before, people knew that from biology, and 1 century later, physicians said, okay, let's see. So I always show this because it's unseen, because it's something that everybody knows. Since Darwin and the physicians, they didn't look at that. But then after 1 century, said, okay, let's consider if in glassy systems we have an ultrametric structure, and we have, and it's really intrinsic. So if you have a glass, you have an ultrametric structure of the replica. So this is the relation.
Okay, let's thank again our teacher. Thank you, obrigado pela presença de todos também, né? Se alguém quiser falar alguma coisa depois, se alguém quiser italiano também, né? Se capite italiano, no? They are all students here. Yeah.
No, I know that.
This two too. This one is from Vanessa. And this
We
one
met in the lab.
Ah, you're right. Ah, okay. Você descobriu. Sabe que eu descobri o problema depois que esse negócio estava pela metade aqui? Ele estava assim, ó. Tava tipo. O problema é esse daqui, estava desencaixado.
Ah, foi isso?
É, ele estava tipo pela metade. Sabe?
Ah. Mas o Brasil é assim, né? Os marcos. Os marcos, tá.
Mas deu certo, acho que deu certo.
Né? Então, eu tirei umas fotos e mandei para vocês.
Ah, obrigado.
Registrar aí.
Agradeço a presença também, professor.